Okay, I guess we can start since it's already time. So thank you everyone for participating in this workshop, and thank you to all the speakers. And first of all, thank you for our first speaker. Our first speaker for today is Alex Graudenzi. He is very recently become a professor an associate professor here in Milano Bicocca. He has been working a lot in cancer in general, mostly cancer phylogeny, and also discovering of oncogenes and pathways for the analysis of cancer. And today he will talk about evolution models and evolutionary signature of human cancer. So thank you again for being here, and please, you can start whenever you want.
Thank you, Simone. I'm going to share my presentation. Can you see?
Yes, we can see.
Okay, first of all, thank you, Simone and Gianluca and Paola for the invitation and for the organization. I'm Alex Graudenzi, working at the Department of Computer Science at the University of Milano Bicocca, plus a bunch of other roles. And first of all, I want to thank my lab, because actually I want to introduce my lab, that it's a multidisciplinary lab, which is, I think, quite important right now, because we have computer scientists, we have bioinformaticians, but also we have statistical physicists and also biotechnologists. And I think this is really important to address fundamental questions in biology in general. We release a bunch of tools for computational biology, bioinformatics, which are typically released on public repositories. So again, this is a starting point. Many of these guys have been involved in the results that I'm going to show you today. And another thing, which is, I think, it's important right now, is that actually it's a nice moment to be in Milan for cancer research. This is just a few of the main institutes working on cancer research right now. And the green dot here is the University of Milano Bicocca, so it's quite well positioned within the map. It's a good opportunity for networking and also for starting collaborations, which is, I think, quite nice right now. And so my talk is going to start from this question that is can the characterization of cancer evolution help improve clinical practices? So in the first part of the talk, I want to tell you something about methods to extract knowledge from genomic data. And but in the second part, I also want to tell you something about other methods to translate such knowledge into clinical tools, I would say. So the first topic will be about a tool that we developed some time ago about the inference of cancer evolution models from longitudinal single-cell sequencing data. And in the second part, we will see how these models can be translated into something that is called evolutionary signatures. So we want to identify prognostic signatures of human cancer. So again, we're going to start from genomic data. We will extract models, and then we will use these models into something that is going to be possibly used into the clinics. So again, the tool is called LACE, Longitudinal Analysis of Cancer Evolution, and we start from this. Probably most of you are familiar with this fish plot. This is a example representation of a tumor evolution. This is an acute myeloid leukemia patient. On the x-axis, we have time. On the y-axis, we have the prevalence of different subclones. So we have different subpopulations characterized by the presence of these different driver mutations. We have a gray clone. Then we observe the emergence of new subclones, the yellow, the purple, and the orange one. After some time, we have chemotherapy, which is good because the tumor shrinks. But as we can see over here, we have relapse. So basically, old clones and new clones emerge and give rise to therapy resistance. So basically, this is bad for the patient. So typically, what we want to do is that we want to exploit sequencing data to identify the evolution, to reconstruct the evolutionary history of a tumor. But typically, cancer biopsies are taken at a single time point and usually at diagnosis. So basically, we can have either bulk or single-cell sequencing experiments, multiple omics, DNA, RNA, or ATAC. And actually, this is good, but actually we have only a single snapshot of the tumor composition. So basically, if we make a cross-sectional experiments over here, we will see just the composition of the tumor over here. So basically, we cannot assess the tumor evolution in time. So basically, it would be good to have longitudinal experiments with multiple time points. First of all, because we can analyze the tumor subpopulation dynamics, we can see the selection phenomena. Also, we can assess the impact of therapies over time. For instance, in this case, and also because this is a side effect, but it's important, but we can increase the statistical power. It's better to have 3 data points than 1, of course. How do we do this in the wet lab? Clearly, right now we have, luckily, we have patient-derived models. So basically, in addition to standard data sets, including human patients, now we can have cell lines, we can have organoids, 3D organoids, we can also have patient-derived xenografts. And this is good for us because actually we can have multiple data points for a single tumor. Okay, so this is quite important. It's, I would say, quite recent in the as a wet lab advancement, but it's really good because for us as data scientists, we can have like access to these newer data sets. But for the computational perspective, we are interested in tumor phylogenetics, meaning that, again, this is an example representation of cancer evolution. We know that cancer evolves through the acquisition of progressive acquisition of driver mutations, in which different subpopulations are characterized by the presence of different mutations. And the general goal of tumor phylogenetics is that we want to reconstruct, sorry, I missed this. We want to reconstruct the clonal evolution of tumors, so the genomic evolution of tumors, by exploiting sequencing data. And why do we want to do this? Because actually DNA alteration are clearly inherited across cell duplications. So they are basically natural barcodes with which we can actually reconstruct the tumor evolutionary history. And again, how do we do this? We can have variant calling. We can perform variant calling from sequencing experiments. They can be bulk or single cell. And when we have variants, when we have mutational profiles, we can get to cancer evolution models. There are many representations of cancer evolution models. We can have standard phylogenetic trees, as in this case. We can have clonal trees. We can have mutational trees. But the idea is that we want to have a representation of the tumor evolutionary history. So there are many approaches that can do this. There are a bunch of tools that are being developed for single-cell sequencing data with single time points. And we developed some of which also in our lab. But again, the problem is that we don't have any explicit temporal information. It's impossible to assess the clonal dynamic in time. And also we need a dock implementation to handle multiple time points. So this is a problem. Also because, to our knowledge, at least at the time, there was only one method for bulk sequencing data with multiple time points, this Calder method. But again, in this case, we don't have the resolution of single-cell data, so we have an intermixed signal coming from the bulk sample. We need to deconvolve the signal, and also it is impossible to map multiple omics on the very same single cells, which is quite important, as I will explain in the following. So we try to fill this gap by developing LACE. Whose primary objective is to use single-cell mutational profiles, mutational data of longitudinal experiments. And with this, we want to reconstruct a proper model of cancer evolution. And this was interesting because actually we, as a side effect, understood that we could also map the genotype to the phenotype for free, I would say, at the single-cell level. I will explain you how in a few moments. So the tool was released already in a couple of versions. It's already on Bioconductor. It's the results of a bigger project of an IARC Cancer Research UK project called Single Cell Cancer Evolution in the Clinic with the Institute of Cancer Research, the Human Technopole, San Raffaele, Politecnico, and a bunch of other institutes. And I'm gonna try to explain you in a few moments how it works. Again, we start from a toy example. Let's suppose that this is the evolution of a tumor. We have different clones characterized by different somatic variants, candidate drivers, I would say. We can see that the tumor starts from this green clone, the clonal trunk, and then we have a subclone, pink one, and another yellow and another purple clone, and the following. What we can do is that we can have patient-derived organoids, for instance. We can have an untreated organoid plus a couple of organoids in which they undergo a specific kind of therapy. And so we can follow the longitudinal evolution of this tumor. And what we can do after is that actually we can perform single-cell sequencing experiments. Again, they can be DNA, RNA, or even ATAC-seq experiments. And so we can isolate cells, and then we can measure the multiple omics on the very same single cells. So this is the input of our tool. Basically, after variant calling, we will have a matrix in which rows represent cells, and columns represent mutations. So basically we will have 1 if this specific cell has got a mutation, degree mutation, 0 otherwise. And we will have 3 matrices, 1 for each time point, and this is the input of LACE. I'm skipping all the details about clearly quality check and of course about variant calling pipelines, but let's assume that you use standard best practices for doing this. And notice that in the current implementation, we consider only single nucleotide variants, but this is clearly an arbitrary choice, I would say. The problem, as most of you may know, is that single-cell mutational profiles are noisy. There are false positives, but especially there are false negatives, meaning that we have a lot of missing data. We have allele dropouts because the coverage is very low. So to make sense of this data, we need statistical inference. In general, this is a general practice. And so I'm going to start from the output of our model, what we want to obtain. And the goal of our LACE is to reconstruct a clonal tree, a phylogenetic clonal tree. And again, in this case, we will have a binary matrix in which rows represents clones, which are called here as genotypes, and rows and columns represent mutation, meaning that the A clone is going to be characterized by the presence of the green mutation, whereas this purple clone is going to be characterized by the presence of the red, the green, and the purple mutation. There is a one-to-one representation from the binary matrix and the clonal tree, which is basically the same representation. So this is the first output. We want to reconstruct this because actually this is the model of the evolutionary history of this tumor. And we have also a second output, which is the so-called cell attachment matrix, in which basically we want to assign each single cell of the dataset to a specific clone, to a specific genotype, meaning that here we will have 1 if this cell is associated to genotype A, and so on and so forth. So basically just it's going to be another binary representation of the cell, the association of each cell to a specific clone. And also this allows us to compute the prevalence of each clone in each specific time point. Then basically we have multiplication. If we multiply these two matrices, what we obtain is that we obtain an expected genotype matrix that is let's say the genotype matrix that we will have if our model is correct and there is no noise in the data. So basically here rows represent cells and columns represent mutations. So again, you can see that basically this is the same dimension of the original data matrix. So the idea, as in standard statistical inference, is that we want to compare our model with data. So basically we are going to perform likelihood estimation. So we have a likelihood function which we can measure the probability of observing this data given the model. As you can see, basically we have the model, this is this G1 expected genotype matrix. Then we have also a model of noise. So alpha and beta represent the false positive and the false negative rates. So actually we can compute the likelihood of observing our data given the model and the model of noise. How do we compute the likelihood? This is a simple product. Actually, we compare each entry of the two matrices, and we will set this value equal to alpha if we have a false positive, 1 minus alpha if we have a true positive, beta if we have a false negative, and 1 minus beta if we have a true negative. Basically, this is just like we have made assumption that I'm skipping right now, but actually we can also discuss about this later. In this way, we can, by multiplying these two products, we actually obtain one single likelihood value for each data point. So in this case, we will have 3 likelihood values for all time points. So we're missing one piece, and then we are done, basically, that actually we want to employ all these time points to compute a weighted likelihood function. Basically, we want to actually have a unique likelihood function. So this is going to be like the way in which we aggregate the different likelihood function for each data point. And basically, we have a weight for each likelihood value. And in this implementation, but this is an arbitrary choice, we wanted to have comparable likelihoods across time points. So basically, these weights are inversely proportional to the number of cells in each data point. So basically, this to say that you want to have likelihood that is comparable across different time points. So basically, this is it. We have a weighted likelihood function. We want to maximize it. We do it with a Markov chain Monte Carlo search on the space of the trees. So we have a preliminary attach move. We have a relabeling mutation move. So basically, we are actually looking for the best model, I would say. And actually, we have different implementation of this algorithm, but actually, this is the main idea. And this is the output. So the output is a maximum weighted likelihood clonal tree, which is quite expressive model because actually we have nodes representing clones with their specific mutations. We can see which clone originates from which other clone. We have parental relations, but also we have persistence relation, meaning that we can see if a clone is still there in the next time point or if it gets extinct. So this is a really expressive representation of the tumor evolutionary history. And I think right now it's one of the best way to exploit longitudinal data. Again, we can compute the prevalence of the different clones and actually represent this history in a quantitative model. We tested LACE against many other tools. I'm not super keen of explaining the results of simulations, but we did a bunch of them. We simulated a lot of simulated scenarios. We made 200 independent data sets. We compared against the state of the art. And we compared over a bunch of metrics, precision recall, but also adjusted rand index to compare like the clustering results. So basically, we are doing better than the others, and which is good. But most importantly, this is very important. Also, we generated also a tool very recently to generate synthetic data sets in a plausible way. Actually, this tool exploits Julia to perform the simulation of a multi-type birth-death process, but over a graph. So basically, we can simulate the cancer evolution model over a lattice, and then we can see the expansion over space, but also we can simulate sequencing experiments. And this is relevant because actually the data that we generate can be used to benchmark models that make use of sequencing data. But most importantly, we wanted to use our model on real data, and actually, at the time we came with the idea of exploiting single-cell RNA sequencing experiments, and actually we were one among the first to do this. And actually, you can have standard full-length single-cell RNA sequencing experiments, and we get to the input of LACE by setting our input metrics equal to 1 if the mutation is present with high coverage, 0 if the mutation is absent with high coverage, or we can leave it as an A if the missing information is due to low coverage. And this is interesting because actually, after us, many others proposed to use single-cell RNA to call variants, to call genomic variants. And this is quite relevant because actually it allows us to have a genotype to phenotype mapping for free, meaning that from the very same single cell, we can have both the information on the genomic alteration and the information on the gene expression dynamics. So this is a kind of vertical data integration, as someone called it, and actually it allows us to define, to identify, to characterize the clone-specific gene expression dynamics by exploiting only single-cell RNA data. So this is quite important in our view. So this is an example application. In this case, we have BRAF mutant melanoma xenograft. We have 4 longitudinal time points. We have single-cell RNA sequencing data. We call variants by exploiting the best practices of GATC, and then this is the oncoprint that represents the mutations that are present in all cells in all different time points. And we applied LACE, and this is the results, which is, I think, a very expressive model of cancer evolution, because on the left you can see that there is the longitudinal clonal tree. We can see actually which clones are present in which time point, which clones originate from which other clones. We can see the mutation that characterize the different clones, and which is really nice because actually these are a very, I would say, quantitative and sound representation of the genomic evolution of the tumor. But also we can map the clone-specific, we can identify the clone-specific gene expression dynamics, meaning that we can see the cell types within each genetic clone. We can see this lower dimensional representation of the gene expression of the cells within each clone. And we can see also, for instance, the cell cycle phases of each clone, of each genetic clone. And this is important because actually it's a natural mapping of genomic to the phenotype, but also because it allowed us to perform, to generate some experimental hypotheses. For instance, that this blue clone, which is characterized by the mutation of this gene, this PRAME gene, is less sensitive to the therapy. Why? Because actually, if you see in this ternary phase, we represent the prevalence of cells within a certain cell cycle phase. we can see that the blue clone continues to cycle during the different time points, whereas the cells in the clonal trunk, they never get into the S phase, meaning that they stop cycling after a while. So this gives us a possible hypothesis to validate, of course, that this specific PRAME mutated gene is less sensitive to therapy, okay? So because it goes on and on by cycling. So this is just an idea of how we can produce the experimental hypothesis. Clearly, as I was saying, this LACE is one of the results of this bigger project, this Cancer Research UK Accelerator project, in which we are collecting and generating many organoids generated from colorectal cancer patients. We have many organoids pre-treatment and many organoids after treatment. And of course, new data analysis pipelines are needed to analyze this data, because actually we, in this case specific for each organoid, we have single-cell RNA, DNA, and ATAC. And so we need an overall pipeline, an extensive pipeline to make the all algorithmic steps that are needed. No, we have preprocessing steps, we have denoising and imputation, batch correction. Of course, we have data-specific analysis, but also we have omics data integration analysis tool. So why do we do this? Because actually we want to get to improve downstream analysis that are comprehensive, that can make sense, and also leverage all the omics data types that are produced. This is just a picture of the organoids that are growing in the lab, which is quite far away from my expertise, but they tell me that they are nice, so I think they are nice too. So the take-home message from the first part of my talk, we developed this method to actually make sense of longitudinal single-cell sequencing data, and the idea is that actually we can quantify the clonal dynamics under therapy, for instance, of patient-derived models, which is quite important. But also, if we want to exploit single-cell RNA data, we can call variants and then retrieve a natural genotype to the phenotype mapping, which is something quite new. And it, again, allows us to have vertical integration for free, which is really important in current biology. Of course, there are many extensions underway. We want to introduce further variant types, structural variants, copy number alterations. We also want to improve the scalabilities, because right now, the method it employs, as I said before, a Monte Carlo Markov change, which is not really scalable algorithm. So we want to improve this, also to possibly apply our method to larger data sets and also to a larger number of variants, and possibly even to spatial transcriptomic data. So this is the end of the first part of my talk. And again, I show you something that is useful, in my opinion, to extract knowledge from genomic data and to derive some models. Now, I want to tell you something about a newer results, about how do we use such models to produce usable knowledge in the clinic somehow. And this is the topic of my second part, the second part of my talk. And it starts from this consideration. This is a quite old review from Schwartz and Schaffer on Nature Reviews Genetics, in which they identified 3 study design types in cancer phylogenetics. One is very common. It's the bulk symbol biopsy case. For instance, the Cancer Genome Atlas datasets, in which we have one biopsy for each patient in the cohort. So what we can do is that we can reconstruct population-level models. But also more recently, we have a multi-region dataset, so multiple biopsies from the same tumor, like in the case of the TracerX consortium by Charlie Swanton in the UK. So basically, we can reconstruct single tumor models. Or, of course, we can use single-cell sequencing data, and also in this case, we can reconstruct single tumor models. Just to be clear, LACE belongs to this category of the methods developed for single-cell sequencing methods, for single-cell sequencing data to reconstruct single tumor models. But of course, countless methods exist for all experimental setups. For instance, if we have bulk single biopsy data, like for the Cancer Genome Atlas example, we can employ one of our oldest tools, which is called PICNIC. It leverages machine learning and a bunch of statistical machinery to reconstruct a single model for a tumor type, meaning that we have a Bayesian network that tells us the most likely trends of accumulation of these mutations for this specific tumor type, which is quite good. It is a robust result. It provides robust results, but it is one of the many tools. Then we can have methods for multiple biopsy datasets, for multi-region datasets. For instance, this is a beautiful work by the group of Giulio Caravagna and Andrea Sottoriva, and it's called Revolver. It employs machine learning and transfer learning to develop to return robust models of single tumors with multi-region data, but also in this case, it is one of the many tools. And why is this a problem? Because actually, this is the result of one of the latest papers published by the TracerX consortium. This is the evolutionary history of, I think, 123 or something non-small cell lung cancer patients. And you can see here that there are beautiful representation of the evolutionary history of all these tumors, okay? But now what? What can all these models be used for? Actually, this question stayed in my mind for some time, because actually, I think this is really beautiful. This is an explanatory model that it's quite nice, but actually, can this be really useful for patients? And this is the question that we wanted to address. Can we use this model to extract prognostic features? And we try to answer to this question with the ascetic tool, which was published some time ago some months ago in Nature Communications. Again, it's a collaboration with other institutes, of course, and it starts again from the inputs. Basically, we can have bulk single biopsies, again, the Cancer Genome Atlas example. And in this case, after variant calling, we will have, for each patient, the cancer cell fraction of each genomic mutation we can consider. So basically here, columns represent genomic alterations, and we have one vector for each patient. So what we can do is that we can order such mutations according to the prevalence, because the rationale is that older mutation will have a larger variant allele frequency, of course, because they are in more cells. But also we can have multi-region data sets, and actually we can use a Revolver, for instance, or other tools to reconstruct the evolutionary history of each single patient in the cohort, in the form, for instance, of mutational trees, and we will have one model for patient in this case. Or we can have single-cell sequencing data sets, like in the case of LACE, or of other tools such as SHITE, in which we can actually have one mutational tree, one evolutionary model for each patient in the cohort. And this is quite important because, as you can see here, we will have one model for each single patient. Well, this is the input of ascetic. We will not consider mutations in this phase, but we will consider models. We will have one model for patient of a given tumor subtype. This is a minor detail, but actually we are considering now the resolution of gene mutated with a single nucleotide variant at any position, but this is an arbitrary choice. So this is the idea. Right now, the input is the model and not the genomic data itself. So this is the overall pipeline. I'm going to try to dissect it very quickly right now. But the first step is probably the most important, because actually we want to aggregate all single models of all single patients. So let's suppose that we have 100 patients here. This adjacency matrix returns how many patients display the red mutation before the orange mutation. So in this case, we will have 44 patients out of 100 that will have the red mutation before the orange mutation in their model of tumor evolution. And only 3 patients out of 100 will display this pink mutation before the green mutation. Of course, you can represent this also as a weighted graph, but it's just like a matter of representation. So again, this tells us how many individual models show a certain ancestor to descendant path. Then we employ some statistical tricks that I'm going to be very quick in the description. Basically, we first derive a ranking of drivers. So basically, we want to understand which mutations occur first before the others. And basically, we do this by employing this algorithm that is called agony, which removes inconsistencies between this graph, this cyclic graph. And this basically allows us to define a ranking of the mutations. So the first level mutation, second level mutation, and so on and so forth. Then we employ some concepts from probabilistic causation, which is a concept that dates back to the 70s, which tells us that C is likely a cause of E if it occurs earlier, and if the probability of observing E given C is larger than the probability of observing E given the absence of C. And this allows us to do what? To retrieve a unique model, the ascetic evolutionary model, which basically is the model that tells us the statistically robust repeated patterns of evolution of a given tumor type, meaning that if you are a patient with this given tumor type, you will gonna most likely start from this red mutation, then you will go either toward the orange mutation or this yellowish mutation, and so on and so forth. This is quite important because these patterns are robust. They are derived from data. So basically, there is no further biological knowledge on this, but they are quite robust because also we employ likelihood fit with regularization and a bunch of other statistical tricks. So we perform another little algorithmic step. Basically, we split the model into pairwise relationships. That basically it means that splitting this model into pairwise relationships, so that we call them evolutionary steps. So you can see here the red towards the orange mutation is going to be a single evolutionary step. And why do we do this? Because actually we want to employ such evolutionary steps as feature to predict the prognosis of a single patient. Again, some of you might be familiar with the Cox regression algorithm. Basically, we exploit survival data, clinical data, and basically we want to understand which of these features are correlated with a better or a worse prognosis. In this case, we employ regularized Cox regression with lasso, but it is a technical detail. So meaning that, again, here we want to see if this couple of mutations are bad or good for the patient. This is a binary matrix, again, in which we will have 1 if this patient displays both the mutation, 0 otherwise. So with what we can do is that actually we will have an hazard ratio, which is the weight of the regression, basically, for each evolutionary step. And then by simple multiplication, like in any regression, we can compute a risk score for each patient. As you can see here, we have different risk score for each patient. And of course, we can cluster this vector into groups, low risk, medium risk, and high risk. And then we can perform the so-called Kaplan-Meier estimate. We can see what is the survival curve for each cluster. You can see here that there is a low risk cluster, a medium risk cluster, and a high risk cluster. Okay, these data are coming from the original data set. So now we know what? We know that we have 3 clusters, and we know which features, which evolutionary steps are more correlated with the better or worse prognosis. And this is it, actually. We call the evolutionary signatures actually the hazard ratio specific of each evolutionary step that is correlated with the high risk, medium risk, or low risk cluster. And basically, so we can know, we can call them also quote unquote biomarkers somehow, which mutation are need to be searched if you want to have a prognostic tool to be used in the clinic. So basically, we will know that if you patient have these 2 mutations and these 2 mutations, your prognosis will be most likely worse than if you have this mutation and this mutation. Again, there is an intended analogy with the concept of mutational signatures that are a completely different theoretical concept, but actually, because actually mutational signatures look for the mutational processes that underlie nucleotide substitutions in cancer, but actually here we wanted to look at evolutionary signatures, meaning that we want to see if there are repeated patterns of evolution that are correlated with prognosis. And again, tests and simulations are fine, meaning that we performed many simulations on a bunch of synthetic scenarios, and we are better than the others. But most importantly, we applied our method to more than 35,000 patients from multiple cohorts. We employed the pan-cancer atlas, so the TCGA datasets, the cancer genome atlas, with bulk single biopsies, whole exome, whole genome, on most cancer types. We employed the MSK impact on a targeted panel single biopsies on 50 cancer types. We employed the one of the TracerX datasets on 100 patients with multi-region whole exome on lung cancer early stage, but also we employed another MSK cohort on with single-cell targeted sequencing with tapestry on 123 patients. And yeah, this is one of the example results. Basically, we have acute myeloid leukemia signatures. We identified 4 different clusters, a lower risk cluster and a higher risk cluster. And here, these are the signatures, meaning that these are the mutations that need to be looked after if you want to know if your prognosis will be better or worse. And this is the model, this is the evolutionary model, so the repeated patterns of evolution that are robustly inferred by the data. You can see that these curves don't overlap, meaning that the different groups of patients actually display a significantly different probability of survival, but also we can see something more interesting, that it's actually if you patient have got only this mutation on this TET2 gene, your prognosis will be much worse than if you have both the TET2 mutation and this other IDH1, IDH2 mutation, which is somehow counterintuitive. Also, this is another example on lung adenocarcinoma signatures. We have three risk groups. We have different mutations that represent different evolutionary signatures. And also in this case, you will have a evolutionary model, and also you will see the stratification of patients in different survival curves. And also in this case, you can see something interesting. It is actually, if you have only the KRAS mutation, your prognosis will be unexpectedly much worse than if you have the KRAS mutation plus the APHA3 mutation, which is something that needs to be addressed by molecular biologists and by clinicians, okay? We validated our data, our results on unseen data. So basically, the results also on cohorts on which we didn't train our model, which is good for us, but this is an important result. And lately, we are actually ending the development of graphic user interface within the Google Summer of Code, because also we want that our tools can be used by the bioinformatics community. So basically, this is the aesthetic vision first screenshots. They're going to be ready, I hope, soon, in which the community will access a usable tool to perform this analysis. So the take-home message from this second topic is this. Actually, we can start from evolution models that can be identified by using any existing tool. There is a plethora of tools that can make the inference of cancer evolution model. We want to exploit such models, okay? From many experimental setup, single cell, bulk, multi-region, single biopsy, or whatever. But then we want to go further. We want to identify robuster patterns of evolution that are associated with prognosis, because we can use such patterns to help clinicians to direct not only the therapeutic strategies, but also the design of prognostic and possibly also monitoring tools. And of course, there are extensions underway. We want to include further variants, also in this case, structural variants, copy numbers, and also other omics data types, and clinical data, and so on and so forth. Again, we want to develop a web-based application on this. And our dream will be like to define a catalog of signatures, of evolutionary signatures for all tumor types, which is somehow inspired to the cosmic catalog for mutational signatures. So basically, this is what is going to be next in our research efforts. And I want to conclude with some advertisement. These pictures are real, incredibly real, but this is going to be like the next edition of the Lake Como School on Cancer Evolution that we organize as the University of Milano Bicocca. So it's going to be like a very good opportunity to talk with both wet lab scientists and dry lab scientists working on cancer evolution. The registration opens soon, so basically if you want some infos, you need to write to me right now, but actually consider this opportunity because it's quite good. It's going to be at the end of May. And yes, thank you very much, and I'm clearly here for questions, if any.
Thank you very much, Alex. Very, very nice talk. Let's see, so we have a bit of delay on the stream, so question may arrive later. To everyone that is listening, please feel free to ask questions even during the presentation. We will then ask them at the end of the streaming. We have both channels to do so, either on Matrix or directly in the YouTube chat. So in the meantime, to see if someone will ask some question, I have one, which is more than a question, it's just a curiosity. So I've seen you mentioned very briefly in your slide, but I also know that it's quite used, I think, in your group, the Julia programming language. So since we have young researchers here that might still be struggling to find what would be their best choice, to write their tool in. I know you used to work in R, and so I would like to know if you made this switch to Julia, and so if you if this could be an help for some of the students, if you think this is a good language to start working on and to learn.
Thank you for the question. Actually, it was a choice, also an attempt choice, I would say, because it was our first experience with Julia to develop this tool for the generation of synthetic data. I think it's a quite useful and a powerful language. We have like, let's say, a general problem is that actually, as long as we try to interact with many libraries that are being developed in the past using single-cell sequencing data for single-cell sequencing data, actually we need to, like to use many languages, also Python, of course, also R, because actually, for instance, if you use ScamPy or Seurat using single-cell sequencing data, you need to interact with these tools. So basically, I would say that we're skipping to Julia, but it's not completely true, meaning that actually we are still using most of the languages. So it depends on your research question. Actually, I think it's a quite good language. It's very scalable. So basically, it can be useful for making like a good computation. But again, it depends on your research needs and research questions. So again, this is quite something that needs to be considered. So actually, it depends on the case. My short answer is it depends on the case.
Yeah, like as usual, right? It
As
depends
usual,
what you want to
unfortunately.
do.
There is
Yeah,
no
thank you.
no, but I mean, it's also important to get towards the direction in which we will get some more standardized, practices. But I think we are not at the point yet.
Okay, yeah. So that was my other question. Yes, do you think it's up to the point to compete with the current? I'd say standard, but yeah, there is really no standard around.
There's
Everybody's
no standard.
writing.
That's the point. I mean,
Everybody's writing more.
there are some attempts of providing guidelines, general guidelines for interoperability, and of course, but actually, I think we are still far away. And also, I think there is a more general problem that I tried to mention quite a few times, which is quite normal and critical. Each computational biology group basically develop his own tool sometimes. So basically, we have an explosion of tools that have been sparsely used, and the few that are most massively used typically come from the larger groups. That doesn't mean that they are the best tools to be used. So basically, something which is clearly, a structural problem, but then it's also another issue to be addressed towards the direction of standardization, I would say.
Okay, thank you very much. The other question is more related to the actual content of the talk, which is how far do you think we are now in towards like prognosis and screening for cancer? Do you think we are at a good point? Is there still work to do? Is it still a big open problem, or are we getting closer to the solution?
It's a very beautiful question. I think the biggest novelty right now is that actually larger and larger data sets are becoming available. So this last part of the talk, which is about basically the identification of biomarkers, I would say, quote unquote, again, is now possible because we have data. We have clinical data which are together with the genomic data. This is something quite new, unexpectedly quite new. A few years ago, it was really basically impossible to get clinic data associated with genomic data. Now we can use even more complex algorithmic tools. We can use deep learning, of course. We can use like many machine learning tools to understand which features, genomic features, are associated to prognosis, for instance. So my short answer is that actually we are. I think that we are going to see very important things, breakthroughs in the next months and years, because now we have data that we can exploit with a lot of computational tools. And before this data, such data was not accessible. So the answer is that probably we're at a good point. Then the second part of my reply, my answer is that actually how and when these discoveries will be translated into the clinic, so will become accessible to the patient, this is going to be probably, it's going to take much longer time, because of course, you need to test your hypothesis, you need to validate the results, and so this needs a longer time, of course. But as for the, I would say, data science part, we are at a very good point in which you have access to hugely larger data sets with respect to the past. For instance, again, we basically analyzed 35,000 patients, which is basically huge. When we made the first analysis on cancer evolution, we were analyzing data sets with 100 patients. I mean, it's completely different. It's an order of magnitude which is different. So basically, this is something really extremely relevant.
Thank you again. Last question, I promise.
Very good.
So you've been in this field for a long time. You've seen the evolution of this field. So what did you think about the recent advancement in AI? Do you think they would fit in this case, not necessarily LLMs or so on, but like general deep learning models? Or do you think there is still space for like more algorithmical and combinatorial approaches?
This is a very nice question. And frankly, I don't have an answer, meaning that now that data sets are becoming larger again, it's quite easy to think that if you apply some deep learning or auto machine learning approach to this data, you're going to get results. Again, the main issue with this is that you want that your models are interpretable, of course, are explainable. So this is the missing part. But we are getting we are improving this also with deep learning. And so actually, I think that as long as explainability improves with deep learning algorithms and they prove to be, efficient and they perform well with this kind of data, there is no reason why, we shouldn't apply them also to this kind of data. And of course, they are doing this, and of course, they are improving the explainability. So it's a kind of a nice run, because actually there is a unique goal, which is like to find, a proper characterization of cancer and proper, to generate the experimental hypothesis on cancer. I also believe that we don't have to be, integralistic on this. We can exploit one method, one approach or the other